I have an integrated research program that looks at, simplistically, 3 different themes: biomarkers, sequencing the genetic landscape of mesothelioma, and looking at immunology in mesothelioma. But all 3 of these themes have the same goal, and that is to improve outcomes for patients with asbestos-related disease. Biomarkers is an umbrella term which can be used to describe different things. For my research, I tend to think of them as biological molecules that can be measured in the blood or the fluid or on the tumour samples. For a biomarker for cancer diagnosis, we'd see that either it was elevated and the cancer was there, or it was negative and there was no cancer. We've been collecting samples, clinical samples, for many years, probably close to 40 years. We've collected blood and effusions, that's the fluid that builds up in the cavities, tumour tissue. We have collected a lot of urine. And these samples we collect not only from the mesothelioma patients, but also from other people, generally who present with different respiratory diseases in the respiratory clinics, but also from other departments within the hospital, and also from the asbestos review program. And so having this collection of samples from different groups is really important when you're testing a biomarker, because you can look at the test and the controls. The biobank is invaluable not only for biomarker studies. Samples that we've collected in the biobank have been part of the international consortia efforts, which have looked at the genetic landscape of cancer. And the Cancer Genome Atlas, the TCGA, have characterised, I believe, over 30,000 different tumours worldwide. And so, in some small way, our biobank has contributed to this project. And none of this would have been at all possible without the patients and people donating their clinical samples very altruistically, with no real expectation that they would get anything out of these studies, but in the hope that future people would be spared the devastation of this disease.